Good morning, everyone. And what we're going to be doing today is looking at the last part of key area 1 of Unit 1, and that is organization of DNA in prokaryotes and eukaryotes. Now, these are terms that we have come across before, and we're just going to remind ourselves of the difference between them. So, a prokaryote is an organism that lacks a nucleus, okay? So, a eukaryote is an organism with a nucleus. So, if we think of examples of each, a prokaryote would be an organism such as bacteria, because we've already learned in Nat 5 that they don't have a nucleus, whereas eukaryotes would be organisms as animals and plants store their DNA in a nucleus. And if you just look here, this is a diagram of a typical prokaryote, such as bacteria, and you can see that there is no nucleus within that cell. Okay, so we're going to look at how prokaryotes store their DNA if they don't have a nucleus to store it in. Okay, so first thing, prokaryotes usually have a single circular double-stranded chromosome. So if you have a look at the diagram, that is this part here, okay, which looks like the red squiggles. And basically, the word circular doesn't necessarily mean it's going to be in a perfect circle. It just tells you that it's not going to be in straight lines, okay? So that is first part of DNA in a prokaryote. However, as it says here, some prokaryotes have a second chromosome which can carry extra non-essential genes, and this is called a plasmid. We've come across plasmids before when we were studying bacteria in Nat 5, and that is the plasmid there, and you can see there it's like a ring of DNA. So that is the two ways DNA is structured within prokaryotes. Okay. Now, if we go on to eukaryotes, okay, and as it says, are organisms with a nucleus containing several linear chromosomes that are tightly coiled and packaged with associated proteins. Right, what does that mean? Well, first of all, the main difference here is that the chromosomes are linear. You see, before in the prokaryotes, they are circular chromosomes. In eukaryotes, they're what's called linear chromosomes. So that's one of the key differences there, okay? And secondly, they are, as it says, they're tightly coiled and packaged with associated proteins, in brackets, histones. So what are those? So these little blue circles here, or they're like, if they're in 3D, it'd be like little balls, okay? They are the associated proteins, or they can also get called histones, okay? And basically the DNA wraps itself around these, okay? So by doing that, okay, basically by coiling around, more DNA can fit into a smaller space, essentially. Okay, now that is quite a common question in exams, okay, asking about how DNA is organized in a eukaryotic cell or a prokaryotic cell. If it asks about a eukaryote, you would need to mention these proteins, okay? So you can either say associated proteins or histones is also acceptable, okay? So that is the two main differences between your eukaryotes and your prokaryotes. As it says here, eukaryotes also have extracircular chromosomes out with the nucleus, mitochondrial and chloroplast DNA. Okay, so we're going to look at diagrams of each. Okay, here we go. So your mitochondrial DNA is found in the mitochondria of both plants and animals. Okay, so if you have a look on this diagram here, you will see that we've got our cell, which could be, this looks like an animal cell, but it could just as easily be a plant cell. Both have mitochondria, okay, and within that mitochondria, they also have some DNA, okay, and this is, you can see it is circular. So they have this circular DNA within their mitochondria. In a chloroplast, which we should know is just only found in plant cells, you can see that DNA can also be found in here. So if we have a look at the diagram, now you don't really need to know anything about any of these, okay? At the moment, all you need to see is the chloroplast DNA, which I've circled, and it's pointing to it here, okay? So you can also find some DNA in the chloroplasts of plants.